It's my great pleasure to introduce my baymate, Xin. Xin did her thesis work at MIT, where she focused on the understanding of nutrient sensing machinery. And in 2021, she joined my Greenberg lab at Harvard Medical School, where she has identified a novel pathway for degrading stimulus responsive, as well as lineage specific transcription factors. This is the work she will present to us today. Xin has won many awards from undergrads all the way to postdoc, and won some of the special ones, including the Arimont Fellowship Fund for Emerging Science Research Award, which she was just awarded recently, and then the Damon Runyon Postdoc Fellowship. Xin has also written several book chapters and presented at many conferences. And one thing that is very special about Xin that I think distinguished her from many, many scientists is that her ability to quickly form a hypothesis just by mining the public data sets without any pipetting. And this is actually the story how she built the foundation for several of her projects rather than one. Where many people say Xin is a rising star, I think that statement is a little bit inaccurate. I think she's already a star in the biological field, and I'm excited for her talk today. So welcome, Xin.
All right, thank you so much, Sky, for the really kind introduction. All right, I'm gonna go ahead. So thank you everyone for joining us today. I'm very excited for have this opportunity to show you our recent work on identifying a novel pathway that can catch many transcription factors for their proteasomal degradation. And also, I will also briefly touch upon like our currently ongoing work, basically showing these pathways having a lot of crosstalk with transcription and also chromatin biology. And so I will start by introducing one of the major players of today's story, the proteasome. So the proteasome is a 2.5 megadalton, very elegant protein complex. It's often called cell's protein trash can. So proteins need to be degraded for diverse reasons. They may already be damaged or misfolded, or they may be part of an invading virus, or they're done with their cellular functions and the cells no longer need them anymore. And the proteasome provides a controlled way to break down proteins into small pieces. In terms of the molecular composition of the proteasome, the active protease sites are hidden away inside of this barrel-shaped 20S core particle shown in color yellow and red. And at each end of the 20S core, an ATP field engine called AAA ATPase, shown in purple, unfolds proteins and feed them into the 20S core for degradation. And on each end of the triple TPase, a large collection of subunits form this 19S lid structure that can recognize substrates and bring them nearby triple TPase for unfolding. And there's a fun fact about the proteasome, which is very relevant for my research, which is that proteasome can localize in both cytosol and nucleus. There are already many past studies have shown this before, and here I'm showing you a new online resource called the Open Cell, where they basically endogenously tagged a lot of human proteins in human embryonic kidney to necessary T cells, and then live image their localization. And from these images shown here, I hope you can appreciate that multiple components from the proteasome's 19S lid, triple TPase, and also 20S core all show very strong and distinguishable localization inside of the nucleus of these hectoansary T cells. And the proteasome has been traditionally viewed to degrade protein that are marked with ubiquitin, which is a special post-translational modifier protein. So this conjugation here has often been referred to as the kiss of death. And a cascade of enzymes ranging from E1 activating enzymes to E2 conjugating enzymes to more than 800 of E3 ligases have been identified by multiple labs in the field to mediate this conjugation. And once protein targets are marked with ubiquitin, they will get shuttled into proteasome for subsequent degradation. And however, since 30 years ago, there have been sporadic papers suggesting some native proteins without modifications like polyubiquitination can still somehow get recognized and degraded by the proteasome. These proteins include ODC, which is a key enzyme for polyamine synthesis, as well as a few cell cycle regulator proteins, and also a stimulus-responsive transcription factor, CFOS. However, how these proteins really bypass the canonical pathway but still can be selectively degraded by the proteasome remain mechanistically mysterious for more than 3 decades. And then basically among them, this transcription factor CFOS really caught my eyes because of a few interesting features it has. So it belongs to the immediate early gene family, the IEGs, which encode stimulus-sensitive transcription factors, whose function is to specifically bind a DNA sequence and subsequently regulate DNA transcription in response to a variety of extracellular signals, eventually impacting gene expression that helps cells to adapt to the initial stimulus. And in the case of FOS, it pairs with the gene family proteins to form a transcription factor dimer and specifically recognize the AP1 DNA motif formed by TGA, XTCA, X is either C or G. And this binding is in response to thousands of upstream stimuli from kind of neurotransmitters to growth factors to immune stimuli to stress. And it regulates also a slew of downstream genes that are cell type specific. In neurons, these genes encode important proteins with key functions like maturation and also kind of plasticity for the neurons as well. So FOS isn't alone in this family. IEGs also include other transcription factor family with diverse protein sequence and structure, including EGR, NR4A families, and MPAS4. And looking at the protein expression pattern of these family proteins, they all share very similar kinetics. So upon stimulus, IEG transcription begins within minutes, causing a burst expression of IEG proteins that display a very rapid decay. And these proper IEG levels directly determine proper downstream gene expression levels, key to ensure cells' adaptations to the initial stimuli. And how IEGs are induced transcriptionally has been relatively well characterized by many labs in the field, including the Greenberg lab. But how IEG proteins, once produced, are degraded so rapidly is mechanistically unclear. And that was the initial question I set out to answer in the beginning of my postdoc. And our exploration in this question led to an identification of a novel proteasomal binding protein that can promote the ubiquitination independent degradation of many stimulus-sensitive transcription factors. And this has been a wonderful collaboration with Chris Nardoni, a graduate student from Steve Allergist's lab at Harvard Medical School. So initially, there are 2 lines of evidence, one genetic, the other biochemical. Both have led to the same protein. So to unbiasedly look for genes involved in IEG stability regulation, we set up genome-wide CRISPR-Cas9 screens. So we use this dual-color kind of system named GPS, short for Global Protein Stability, which allows the stable expression of DS-RED as an internal control protein, together with the GFP-fused IEG protein from the same bicistronic mRNAs. Therefore, this green to red ratio measured by flow cytometer can serve as a measure of the relative stability of GFP-fused IEG proteins. So you can imagine in this format of CRISPR-Cas9 screen, if we knock out a degrader toward IEG proteins, this ratio is going to go up. So we basically performed a screen for both EGR1 and FOSB, 2 very different IEGs with diverse sequence and structure. And here are the 2 plots showing the screen results. Left is for EGR1 stability regulation, right is for FOSB. And quite surprisingly, the top hit from both of these screens was the same gene, MIDN, along with a few other proteasomal components. So we validated the screen result, and this is a Western blot, shows the endogenous EGR1 protein expression and FOS protein expression levels. They both are dramatically stabilized in MIDN knockout technoid T cells compared to the wild type. And then when we reintroducing back the cDNA of this MIDN, it's sufficient to promote the degradation of both of these IEG proteins even below the baseline. We have also validated this phenotype in multiple other cell lines, including depolarized neurons and fibroblasts. And meanwhile, we have also measured the IEG mRNAs in these cells and observed there was no change upon the menolan basically when you try to get rid of menolan or when you overexpress it, indicating this amidine's degradative property to where IgG proteins are on the protein level. So on the other hand, the biochemical evidence came from mining an unbiased kind of data set published by Steve Gicke and Wade Hubbard's lab at Harvard Medical School. So this data set's name is called Bioplex, which is a protein-protein binding data set. So I found in this data set that several different families of IgG proteins, EGR1, FOSB, and NR4A1, when stably expressed in HEC23 T cells, can co-immunoprecipitate the protein product encoded by this MIDN gene. So this MIDN gene can encode the protein named menolan. And then not just the IgG proteins can bring down this menolan protein, also a proteasomal component called PSMD2 also does the same, really suggests a potential biochemical association among IgG proteins, this menolan protein, and the proteasome, which we validated biochemically by co-immunoprecipitations. So here's like a brief summary. So we found this protein menolan, it biochemically binds with IgG proteins and the proteasome, and functionally it promotes IgG proteins degradation. So if we inhibit the proteasome's degradative function, menolan can no longer promote the substrate degradation anymore, really indicate that the proteasome is required for menolan to work as an IgG protein degrader. In contrary, when we inhibit the polyubiquitination enzymatic cascade by small molecules as well, there is no impact on menolan-sensitive kind of IgG degradation process at all. And when we mutate all lysine residues on menolan target protein, this IgG proteins to arginine, which can block the lysine kind of polyubiquitination events, this process also does not affect the function of menolan. So these evidence together strongly suggest the polyubiquitination pathway is likely not involved, but the functional kind of proteasome complex is actually necessary for menolan to work as a degrader. And also quite interestingly, just like the IgGs, menolan's own transcripts are also induced by various upstream stimuli. This is measured by qPCR. And then, but it's to much smaller fold changes compared to classic IgG genes like EGR1 and FOS. So you can see here in primary cortical neurons, this MIDN transcript can be induced by potassium stimulation, which mimics the depolarization of neurons. And also the growth factors can induce this MIDN transcripts as well in fibroblasts. So conceptually, it makes a lot of sense to actually upregulate the menolan protein to keep up with these transiently induced substrate proteins. Transcriptionally, we also have obtained chromatin binding evidence showing that multiple IgG proteins can actually go to the menolan gene locus to induce its transcription as well. And so far, what I've shown you is multiple IgG protein from different families can be regulated by menolan. And this begs the question whether menolan can also degrade other type of substrate proteins beyond just IgG proteins. So to unbiasedly explore this question, we perform a cDNA-based screen to identify additional menolan targets. So we use the GPS kind of system again, but instead of fusing IgG proteins to GFP, we fused barcoded 12,000 human open reading frames to the GFP, comprising a cDNA library. And then this library was stably introduced into the MIDN knockout HEC23 T cells. So each cell here expresses one human WRF. And then we compare the protein stability of each human WRF in two cell populations, one completely lack of menolan, and the other was menolan overexpressed to look for human WRFs that are regulated by menolan. So the cells expressing human WRFs whose stability decreased upon menolan overexpression, like the cell shown here, are cells of interest. And we can sort out these cells and measure the stability change of each human WRF by sequencing and quantifying the barcode. So the result of the cDNA-based screen is shown here. X-axis shows all human WRFs from the library that we can identify from the final sequencing. Y-axis shows the stability change comparing the two cell populations. So more negative the score is, more regulated this hit is by menolan overexpression. So really, to our surprise, and very excitingly, 5% of the library, which are 500 human wolves, are destabilized significantly by menolan overexpression. And about half of these 500 proteins have experimental data showing they can be localized inside of the nucleus. And transcriptional kind of regulator protein that can bind with DNA are also highly enriched. So this screen really suggests a broader spectrum of menolan substrates beyond just IEGs. Here I'm showing you several more examples that we have validated from the top 500 hits. And you can see that FOSB, which is our positive control, scores almost at the top, along with many other proteins localized in the nucleus involved in transcriptional regulation. CBX4 is another positive control. It's a polycomb kind of component. It has been reported to be degraded by the fly homolog of menolan, Stuxnet, in 2016. And also many cell type specific transcription factor also score, like this IRF4 protein, which is important for T cell and B cell homeostasis, and also a key oncogenic driver for myeloma. So we have validated its menolan regulation in Ramos B cells. And then you can see in the population knockout of MID and endogenous IRF4 protein go up. And when we overexpressing more menolan, its level goes way down. And then consistent with many targets of menolan being nuclear proteins, menolan itself is also primarily localized inside of the nucleus, shown by this immunofluorescent staining of menolan. And so now we have expanded the target repertoire of menolan to many nuclear proteins. But how does menolan really mediate its interaction with the proteasome and also the substrate proteins? So to answer this question, I use AlphaFold to predict a structure for menolan, and we found 3 conserved and structurally defined domains. And by biochemical experiments to validate the functions, we figured out that the C terminal of a helix C, showing color green, is used to stably bind to the proteasome. And also for the substrate binding, this yellow-pink domain we named catch is employed. And to really figure out how this catch domain works to bind with so many different proteins, we employ AlphaFold structural prediction again. So we fed the AlphaFold structural prediction tool the menolan sequence together with its target sequence. So the left is the prediction for the catch domain itself. And on the right, you can see an unstructured region within the menolan target protein IRF4 adopts this beta strand conformation after binding to menolan's catch domain, completing this 5-stranded beta sheet tertiary structure. And we were really, really excited about this prediction. So then we went ahead to validate it by making the deletion, also mutation from both the target side and also the catch domain side. And this mode of interaction indeed can be validated. And then we took the 500 top hits from the cDNA screen that I showed you earlier and performed AlphaFold structural prediction for all of them individually with menolan. And what's super exciting is that 40% of them yielded promising beta strands. That's about 200 of beta strands that we found. And I'm showing you 8 more examples here. So they insert right into the catch domain. And this mode of interaction can really try to mediate, so many different target proteins interaction with menolan's catch domain. And this really suggests the mode of interaction is indeed a general mechanism, and we also have validated many of them. And to further validate this interaction mode, we team up with Serrano and Huck from the crystallography core at Dana-Farber Cancer Institute. So we purify the EGR1 beta strand fused to the menolan catch domain, this fusion protein, from bacteria. And here I'm showing you the AlphaFold predicted structure of this fusion protein. Yellow is the EGR1 beta strand, pink is menolan catch domain. And then basically we purify this protein from bacteria and then concentrate it and then form a crystal out of that. These are how the crystals look like. And from them, they basically yielded a 2.5 Armstrong crystal structure, which we further modeled in the right. And you can see the red is the EGR1 beta strand, blue is menolan catch domain. They look very similar to the predicted structure on the left. And in fact, when you overlay them, they look almost identical. So indeed, this beta strand insertion mode of interaction can be backed up by real structure data as well. So after binding with a proteasome and also a substrate protein, menolan will use this third very conserved domain, showing the blue, which looks like a kind of ubiquitin protein, to really promote the degradation of the bound substrate. So our result really converged into this working model. Both menolan and its many target proteins can be induced by diverse upstream stimuli, and they localize inside of the nucleus, and menolan really can serve as a proteasome's hand. It binds with proteasome quite stably via this conserved alpha helix C and adopts unstructured regions of the substrates to beta strands via its catch domain and promotes the substrate degradation in the UBL-dependent manner. So this menolan-mediated kind of degradation mode is very non-canonical, so it only requires the substrate protein having unstructured region with beta strand forming property. So there's no PTM seems to be required for this interaction, which strongly is in contrast to the canonical pathway that is centered around the polyubiquination modification via a complex enzymatic cascade. So there are a few ongoing and future directions are kind of mentioned here. In collaboration with Susan Shell's lab at Harvard Medical School, we are characterizing how structurally this really works, how menolan structurally bind with proteasome to degrade substrates. And also in collaboration with Ben Ebert's lab at Dana-Farber Cancer Institute, we're exploring the crosstalk in between the menolan proteasome pathway with transcription and chromatin events, the transcription events happening on chromatin. We actually see this very surprising finding where this menolan proteasome complex, which is a 2.5 megadalton gigantic complex, can be recruited to chromatin to regulate the transcription factor that are actively bound on chromatin. So we're very excited to go ahead to figure out what's the meaning of this interaction. Why does the degradation need to happen on chromatin? Can that have other roles beyond just a simple degradation event? And also, super excitingly, I've made a mouse model to explore the in vivo functions of this pathway. And I'm currently on the job market and I will soon begin my own lab. So if you're interested in working with me, send me an email. And here's my email address. So with that, I would like to thank everyone who has contributed to this project. I want to thank Mike, who's my wonderful advisor, and also everyone from the Greenberg lab for their help and support along the years. And this menolan story is in collaboration with Steve Allerger's lab with huge amount of help from Chris, Nolan, and Aoyue. And also current structure, the cryo-EM structure work is done together with Susan Shell's lab and with help from Jingjing Matthew and also Millen. And I'm also collaborating with Ben's lab together with his very talented graduate student, Justin Roger. And also the crystallography work is done together with Huck and also Serrano. And the mouse line I made together was the core from Harvard Institute with Lin Wu. And also I would like to thank my funding sources, the Leffler Center, the Iranian, and also the Aramon Fellowship Fund. And yeah, thank you so much for your attention. I'm happy to take questions.
Thanks, Xin, for the wonderful presentation. And if you are looking for a position, I will definitely reach out to Xin. And if you have any questions, please feel free to post it in Q&A box. Jackson, I think you can start asking your question.
Yeah, Xin, this is such amazing work. It's really cool.
Thank you.
I'm curious if you've looked at in your menolan knockout cells, do these cells just have like an unattenuated response to transcriptional signals or to stimuli? Like, do they fail to silence the response after they start it?
Yeah, that's a great question. So we're currently in the process of trying to figure out more the cellular-based type of phenotypes. So if you basically do an RNA sequencing, trying to compare the wild-type cells to the menolan knockout cells to the menolan overspread cells, there are basically thousands of genes, their level are changing quite dramatically. But so far, we can still make the clonal KO in the HEC-transferred T cells, which means that at least for cell survival, having menolan being there is not really required. But my speculation is really based on a lot of target protein by this pathway involve a lot of key transcription factors that are important for differentiation and also cell fate transition. So I'm like curious to make the kind of knockout in a stem cell state and try to differentiate them to see whether this pathway is actually involved in the cell fate transition process, because there are many transcription factors need to be turned over, turned off, or turned on for that differentiation process to happen. Yeah, so we haven't really seen too much of a cellular phenotype yet. That was, yeah. Thank you.
Great. So we have several more questions. Due to the time limit, we'll probably move on to the second speaker, but please feel free to type your question in Q&A box so that Xin can answer your question and in the second half of the talk. Thanks, Xin. And
awesome. Thank you.
Yeah.